My name is Julie Lewis, and I'm a PhD candidate studying tick vectorid bacteria and epigenetics using clinical samples from Lyme disease and Morgellon patients, as well as infected cell line models. But today, the research I'm presenting was from my master's degrees that is part of the ongoing Lloyd tick bank study at Mount Allison University in New Brunswick, Canada, the same lab that gave a few presentations yesterday. Okay. There exist a lot of Borrelia species. Here are just a few listed. But to assess the Lyme disease in our study, we looked at the primary Borrelia species responsible for Lyme disease, Borrelia burgdorferi. As a side note, we published the second objective of my master's project, the detection of Borrelia biseti ticks for the first time in New Brunswick, using the primers that Samantha mentioned to you yesterday. But I don't have enough time to talk about that today. So Borrelia has a very unique genome. They have a linear chromosome, which is already rare for a prokaryotic cell. And they also have more than 20 linear and circular plasmids. One of the two genes targeted in our study is a chromosomal gene, and the other is located in the plasmid. Sorry, I kind of have to go back and forth here. In Eastern Canada, the main tick species responsible for transmitting Lyme disease is Ixodes scapularis. The distribution of Ixodes scapularis ticks in Canada, collected between 2004 and 2012, is represented by dots on this map. The Lloyd lab study documents the distribution of ticks and Borrelia burgdorferi distribution in the three Canadian maritime provinces, highlighted in red here. So there's one that is endemic, another that is emerging, and the third at no or low risk area. What is more interesting, however, for us was to go in more detail with New Brunswick, as we received a lot of ticks from there, and it's where the study is being conducted after all. As climate changes, the ticks are predicted to establish northwards, as these maps of New Brunswick show over time for the year 2020, 2050, and 2080. And these pictures are shown with two different prediction models. So that's why there's a double of each. Ticks have a very limited dispersal range on their own, but they have many different hosts on which they can travel long distances. And these are all the hosts that from which we receive the ticks in our studies. Birds play an essential role in tick dispersal. Each spring, an estimated 50 to 175 millions of I. scapularis are being dispersed into Canada by migratory birds. And New Brunswick is near the junction of two flyways, one that goes within the American continent and the other that reaches across the Atlantic Ocean. So there is a dispersal of ticks on a local, regional, national, and global scale, which fits with the One Health initiative perfectly. For this study, ticks were donated by veterinary clinics and the general public from 2012 to 2017. These ticks were processed at Mount Allison University. I put a little picture of the campus there for you guys. These were the ticks that we got in one day on November 2014. The participant filled out a form to provide us with the information such as collection date and location, travel location if there was any, tick host, and so on. I have seen many different ticks, and let me tell you, I have selected the best looking ones for this presentation. There are 3 developmental stages. So you have the larvae, the nymph, and the adult. To distinguish between tick developmental life stages, you need to look at things like the number of legs and the presence or absence of the genital pore in the adult tick. To differentiate between the tick species, the shape and the pattern of the tick shield is used amongst other morphological characteristics. These are the 3 tick species that were mostly collected in our studies. Also, the size of the shield is used to determine between female and male ticks. So in a female tick, the shield is much smaller than the males. And in these same 2 last pictures I just added, we see a tick that has fed for a few days and one that has not. DNA was extracted using the following steps to liberate the DNA from the cells. Oh, there. And to precipitate it out of solution. After, we have the DNA of the tick and the DNA of the microorganism that are infecting the ticks, if there are any. And then we target the Borrelia DNA as shown in the next slide. So the tick infection testing was done by nested PCR, where we amplify a fragment of DNA from 2 Borrelia genes. And all the infected tick data that I will be mentioning from here onwards were found using these 2 genes, these 2 Borrelia genes, so OspA and flagellin B. The number of scapularis tick generally increased over the course of the study. In this graph, you can see the proportion of ticks that were infected and uninfected for each of the years. In total, we tested 2,953 ticks from New Brunswick, 493 ticks from Nova Scotia, the neighboring province, and 4 ticks from Prince Edward Island. Tick species other than scapularis were also found in our study, and they transmit various diseases. So I just wanted to show that quickly as a side note. The other category includes a rabbit tick, 1 brown dog tick, and 2 mouse tick, whereas the unknown tick species category were specimens that were too badly damaged for species determination. Now, ticks data have been arranged by seasonal distribution. Ticks were received throughout the year, but the majority of them were collected in the months of May, June, October, and November, which corresponds to the tick spring and fall tick seasons. Between 2012 and 2017, 79 infected scapularis ticks were found on humans, 341 were found on domestic animals, and 23 were found on wild animals, unattached, or on unknown hosts. Our study at the Lloyd lab adds to previous studies by providing a bigger sample size, giving us a better idea on the tick infection rates for Borrelia burgdorferi. Province-wide infection rates was found to be within the range of previous studies on the same subject. But things start to get more interesting when we take a more detailed look within the province. So this is the current map on public health's websites. And this map here is from the Lloyd tick lab. So these are the data through the years with different colors assigned to each of them. We can see that the ticks are up the coast and further northwards than the risk areas indicate. To conclude, we found there was a correlation between tick number and known high-risk areas, but there was no correlation with the tick infection prevalence. This indicates that risks do occur outside of known endemic areas, and accurate and timely information on tick and Borrelia burgdorferi distribution are essential for keeping the public and medical communities informed of risk and prevention efforts as the disease emerges. Here are some references that I used, and here, there. And I will let you go with some pictures of New Brunswick in case you feel like traveling, but you can't with COVID. I just wanted to thank you guys for organizing this conference and inviting me. It's been a while since I've been to a conference. It was way overdue. And I will take questions if there's time.